And so then, sorry, I'm just checking my timer again. And so this is very interesting because A2A receptors are known, at least in cortical synapses, to induce glutamate release. They are very much they're highly expressed in the striatum areas, and mainly they interact with D2 dopamine receptors. But in cortical areas, they are mainly presynaptic and they induce glutamate release. Although this mechanism is not totally understood, we know that whenever you activate the receptors, you induce glutamate release. And also canonically, or their canonic downstream pathway is through cyclic AMP and then CREB actions. Also, we know that they are able to phosphorylate some of the voltage-gated calcium channels, this time the presynaptic, so the N-type calcium channels. On the other hand, these receptors, as of course this audience does not need to know, they are blocked by caffeine. So, but the important thing is that usually their expression is very low in cortex, in the hippocampus. And if you look both in animals and humans, you really see a very low expression. So why in aged synapses, if you block them, they are so important to normalize the synaptic dysfunction. And so indeed, if you, for instance, if you block A2A receptors and if you give an oral, an A2A antagonist to animals, you don't see many differences. And this is the Maurice Watermay, so a memory test that we do. And so we don't see many differences in the animals that were receiving vehicle or animals receiving, for instance, KW6002, which is a selective antagonist for A2A receptors. Not even you see much of an effect on either LTP, long-term potentiation, or long-term depression when you talk about young animals, as you would expect, since the expression is very low. So what we've seen is that to try to understand what is happening here, we transfected neuronal cultures with an A2A construct that links to a venous. So you'll see venous signal whenever you have transfected cells. And so this is, you see two cells here that are transfected and are green here, and this is the calcium signal. And then what we do is that we use a calcium sensor to understand what happens to calcium. And what we've seen is that when you activate A2A receptors with CGS, which is a selective ligand, you have an increase in calcium in the transfected neurons that you don't see in the untransfected neurons. And then this is just the positive control with ionomycin. And more importantly, this is blocked if you block an NMDA receptor. So somehow this tells you that there is an entry of calcium mediated by A2A receptors that is dependent on, that is through NMDA receptors. And this is just a video for you to see what I'm saying. I hope you can see it. But this is a non-transfected cell. And if you look into a transfected cell, you see a much more calcium increase in the neuron that in which the A2A receptor is being activated. So when we were looking into aged animals, one of our PhD students remarked something that was really interesting, is that when you do a cognitive test, this time just a short-term memory test called Y-maze, and usually animals, actually what it does is that animals go through two arms and they prefer always the old arm versus the new arm versus the old arm they already visited. And so this is a young animal, which the way it should be, so they prefer the novel arm compared to the other arm. And an aged animal, they cannot really discriminate. And we use this as a fast screening for hippocampal dependent memory. So what we remarked is that in these aged animals, we had a subset of animals that were aged impaired, so they were really old. And some animals, they would behave like young. So we could not, they could still discriminate and they will behave like young like animals. And then more importantly, we could correlate this with the percentage of LTD dysfunction I've shown you in the beginning. So somehow there were some synaptic features that were correlated with this different trajectory. Some animals, although they are old, they are not as impaired. And more importantly, those aged impaired animals, when they were treated for a month with an A2A selective antagonist, we could normalize the cognitive impairment, which was very interesting. And so this was very interesting, as I told you, that's something we are working on. More interesting was that a cohort of human subjects in which the investigators they had access to the post-mortem cortical brains, they associate, and then they did some proteomics, they could associate cognitive stability here in pink with increased in synaptic proteins related to glutamate synapses and also with mitochondrial activity. And this proteomic signature came mainly from neurons, whereas fast declines, the subjects with fast decline, this was associated with decrease in proteins related to inflammation, myelination, and apoptosis. And so somehow the integrity of glutamatergic synapses is linked to a more stability or cognitive stability over time. So we also did some work on human brain, and we found out that in human cortex, you really have more A2A receptors here in brown, and you can see more A2A receptors either in aged brain and also in Alzheimer's disease brain. If you look at the protein, you can see here a young subject. Young, when I say young, is 25 to 35 years old, and an aged subject over 65 years old, you see a large overexpression of the protein, and we see even a more striking effect in AD-derived brains, and you can see the quantification of this. And so what we thought, because this was hard to study and we need really good models to study A2A receptors, and as I told you, the expression is so low in young animals that we could not really study. So we created a transgenic model in which you induce overexpression of the A2A receptor under the CAM kinase 2A promoter, which is mainly expressed in hippocampal and cortical areas. And you can see here that we have really an overexpression of the receptor, mainly in the hippocampus and cortex, as compared to wild types, whereas the other brain areas, striatum, hypothalamus, cerebellum, and peripheral areas like spleen and adrenal glands had no significant overexpression. So we used this model to understand a bit better what was happening in aging. So the first thing we remarked is that the A2A receptors seem to be mainly presynaptic in the CH3-CA1 projection. So they were mainly going from these projections to in the terminals to the CA1 synapses. And you can see here we did some fractionation in which A2A receptors are mainly in the non-PSD, so the non postsynaptic density fraction, and more in the SNAP21, so mainly the presynaptic fraction. And then, of course, the first thing we did was whether these animals also had cognitive deficits that we see like in aging, and actually they do. So if you do a Morris-Water maze, you see that the learning curve for the Morris-Water maze is decreased or is impaired in the transgenic animals, as you can see here in red. And then we did individual recordings by patch clamp to understand what was happening in terms of function of this neurons. And what we remarked was it's a purely presynaptic effect, and we did this with paired pulse ratio. And these effects in paired pulse ratio were completely blocked if we block the receptor. And also we remarked that we had really a difference in the NMDA receptors. So the channels were more activated, although we had no alterations in subunit composition or intrinsic excitability of the neurons. And then, so again, these transgenic animals have this this LTD to LTP shift I've shown you in aged animals, But more importantly is that if we increase NMDA blocking, so if you start by titrating NMDA and if you increase NMDA receptor blockade, you normalize this to depression again. So it's an effect that it seems that it's not structural, because acutely you can revert this effect in hippocampus lysis. Also, one thing that is important for the aficionados is that one of the reviewers actually asked us to show if this was a calcium gradient, which it seems so. So in our protocol, if you see here, then we did stimulations with different frequencies. So this is the frequency used, and within this frequency, in controls, you have a depression, and then you have a stimulation in transgenic animals. But then if you decrease the frequency, you get the opposite effect. So it seems really that depends on the calcium intercellular concentration is different in aging. And so there's why we call it synaptic plasticity dysfunction. And we do not say whether this is LTD or LTP. It's really a threshold problem. And of course, for the Purine Club, I had to bring in also, we of course gave caffeine to these animals, and caffeine was able to completely normalize this LTD shift. Okay, so in some work before, we had shown that activation of metabotropic glutamate receptors was indeed mediating this overactivation of NMDA receptors by phosphorylating FeN, and FeN would phosphorylate NMDA receptor to be subunit in a previous study. And we thought, why a presynaptic receptor would elicit a postsynaptic effect? And we thought that maybe metabotropic glutamate receptors were a good mediator. So that's what we tested. Mainly our hypothesis that maybe A2A receptors are inducing glutamate release, more glutamate release than sensed by metabotropic glutamate receptors that in turn would phosphorylate NMDA receptors. And what we did is that when we blocked mGluR5, you could really normalize this effect. And so if our data may explain this apparent paradox in aging NAD, which is NMDA receptors are seen to decline. So whenever people do batch analysis, they seem to decline, but NMDA antagonists, such as memantine, have beneficial effects in some of the patients. So it may be that the expression does not really express this overactivation. And our conclusion, I'm just going to finish saying that somehow the stability of glutamatergic synapses seem to be linked to cognitive resilience. And also maybe this metabotropic GluR5 NMDA recruitment that I've shown you that is mediated by A2A receptors may underlie calcium dysfunction in age-related cognitive deficits, which will be great to be able to really validate in human brains or functional, which is not a possibility yet. And so with this, I want to thank my group in Lisbon, particularly Mariana, that did most of this work, and Joanna, and of course our facilities and funding, and all our wonderful collaborators all over the world. A lot of them are Purine aficionados. Thank you so much. You are muted.
Thank you, Luísa.
Thank you, Luísa, for this very nice talk. Thank you, Ahmed. I didn't see. So I suggest we move to the next speaker. And feel free to ask questions in the chat. We will ask to the speaker after the last talk. So Jean, it's you on stage.
Can you hear me?
Perfect.
Okay, good. Thanks, David. Thanks to the organizers for giving me the opportunity to share with you some of the work we have done on some of the work we have done in the lab. So today I'll talk about ectonucleotidases, especially the nucleoside triphosphate diphosphohydrolase. This is enzymes that we have identified and cloned and characterized. And I want to show that how these enzymes can regulate P2 receptor signaling. It's not only necessarily the most obvious effect to hydrolyze nucleotides, but it can regulate nucleotides in different ways. So that's what I would like to talk about today. Okay. Stop. As you all know, nucleotides are released in the extracellular environment by several different mechanisms. And once the ATP or UTP is outside the cell, as you know, they can activate 7 ionotropic P2X receptors and 8 P2Y metabotropic receptors. So you all know that. These molecules can be hydrolyzed by ectonucleotidases. They will produce the molecule with 1 phosphate less, so 8-DP and UDP. And these molecules can activate different type of P2Y receptors, which is also very important. And these molecules can be further hydrolyzed by ectonucleotidase, the same or different ones. And AMP is hydrolyzed by ecto-5' nucleotidase or alkaline phosphatase to generate adenosine. And as you know, adenosine is very important to activate 4 types of adenosine receptors. And it's very important to regulate the concentration of these guys here, because the activation of P1 receptors most of the time trigger an opposite effect to what is triggered by P2 receptors. For example, P2 receptors are pro-inflammatory and adenosine is an anti-inflammatory. P2 receptors will stimulate platelet aggregation, while adenosine is an antiplatelet. And the smooth muscle cells, P2 receptors will be, when activated, they will induce contraction. But when adenosine receptors on smooth muscle cells are activated, it will induce dilation. So what we're interested in the lab is mostly these guys here with the P2 receptors. But as this all goes together, of course, we have to work on 5' nucleotidase and adenosine receptors as well. So we have a focus on anti-PDAses, as I mentioned earlier. And we have a focus on inflammation, the role of these guys in inflammation, inflammatory bowel disease, and on smooth muscle cell contraction. These are the most dominant functions we are studying in the lab. So one way to that anti-PDAs can regulate ectonucleotidase and anti-PDAs can regulate P2 receptor activation is obviously by the hydrolysis of nucleotides. And here I show the hydrolysis of ATP in yellow by anti-PDAs 1, anti-PDAs 2, 3, and 8. Those are the enzymes that are the most important ectonucleotidase to hydrolyze nucleotide at the cell surface in physiological condition and physiological pHs. I'm not talking about anti-PDAs 4 to 7 because they are mainly associated to intracellular membranes and they are not expected to hydrolyze nucleotide at the cell surface. So as you can see, ATP is very well hydrolyzed by all of these enzymes. And in the case of anti-PDAs 1, an ADP is produced, but is rapidly hydrolyzed, and there is a rapid formation of AMP. With anti-PDAs 2, anti-PDAs 2 is not so good to hydrolyze ADP. So we have an accumulation of ADP with not much AMP forms. So that means somehow that anti-PDAs 1 could block any P2 receptor activation, but anti-PDAs 2 could favor the activation of some receptors responding to ADP, such as P2Y1 and P2Y12. And anti-PDAs 3 and anti-PDAs 8 are in between of anti-PDAs 1 and 2 as they hydrolyze ADP, but not as efficiently as anti-PDAs 1. So this could suggest that in presence of these enzymes, we could have a transient activation of receptors responding to ADP. Uracil nucleotides are also very important for some P2Y receptors, P2Y2, 4, 6. So the hydrolysis of UTP is also important. And as you can see, AUTP is also a very good substrate as for ATP for all of these enzymes. It's hydrolyzed very rapidly by all of these enzymes. The difference is that UDP is not as efficient agonist, as efficient substrate than ADP. So UDP is hydrolyzed, but a little bit slower compared to ADP. So that could suggest that the P2Y6 receptor that is activated by UDP could be transiently activated by some of these anti-PDAs, but would be directly activated with the help of anti-PDAs2. So that's what these results suggest on the biochemical and enzymatic point of view. Also, an important function of anti-PDAs is that normally it's ATP that is released from cells. So the hydrolysis of ATP is important to generate adenosine, but for this we need also 5' nucleotidase, because anti-PDAs hydrolyze up to the substrate of 5' nucleotidase, and 5' nucleotidase is the enzyme that will produce adenosine from the complete hydrolysis of ATP. And here, that's what we did. We used anti-PDAs1 in the same time as CD73, ecto-5' nucleotidase, combined hydrolysis of ATP, and for each combination here as well. So with anti-PDAs1, of course, it hydrolyzes ATP, it produces ADP, which is hydrolyzed, and then we have a rapid formation of AMP, which is then hydrolyzed by 5' nucleotidase, which leads to a rapid generation and complete turnover of ATP to adenosine. And with anti-PDAs2, there's no adenosine produced because anti-PDAs2 is not so good to produce AMP. And anti-PDAs8 is not as good to hydrolyze ADP than anti-PDAs1. So the AMP produced is delayed. But the formation and then adenosine appears, but a little bit later. So this gives gaps to be able to stimulate some receptors or adenosine receptors. So with anti-PDAs1, you would expect to have a rapid conversion from P2 to P1 activation. And with anti-PDAs8, this would be delayed, anti-PDAs3 as well. And this would not be possible in the presence of anti-PDAs2 alone. But this is not the only important thing here, is that ATP and ADP are also two potent inhibitors of 5' nucleotidase. So that means that anti-PDAs1 is not only providing the substrate of 5' nucleotidase rapidly, in the same time, it's also removing two very potent inhibitors of 5' nucleotidase. So that allows the enzyme and 5' nucleotidase to be very active very rapidly. And in the case of anti-PDAs2, you don't have substrate or not much, and you have high concentration of the inhibitors, so it's almost impossible to get adenosine produced. So that would be important, probably important. But again, this is a suggestion by the biochemistry and osmology, so this is not a proof in vivo. So another thing that can obviously help to have different function for these enzymes and to regulate different biological function is the expression of these enzymes. And here, this is just to give a rough idea about PCR, that the expression of anti-PDAs1, anti-PDAs2, 3, and 8, as you can see, the expression is different from tissues and cells. But what is most important is the expression, the cellular expression of these enzymes. And I want to show you just two examples, just to give an idea that the distribution of these enzymes is very different from for each type of anti-PDAs. So here we have sections of livers. We have a portal tract, triad here, and as well here. And as you can see, anti-PDAs1 is highly expressed on the endothelial cell with this Ross precipitate, is very highly expressed on endothelial cells of an artery. Smooth muscle cells also express the enzyme. Capillaries surrounding bile ducts are also expressing anti-PDAs1. And the sinusoid with endothelial cells in the tissue may be a bit difficult to see here, but the sinusoid and endothelial cells in the tissue are also labeled by the enzyme. And the endothelial cell of the vein as well is stained. Anti-PDAs2, in contrast, is not expressed by endothelial cells. It is expressed by cells, fibroblasts underneath the vein. It is expressed by adventitial cells surrounding arteries. And it's also expressed by a type of fibroblast that we have identified as a kind of new type of fibroblast in the portal tract that NTPDase2 is also expressed in fibroblasts surrounding bile duct, which is important for communication with bile duct cells. And as you can see, NTPDase8 is expressed in bile calaniculae in the hepatocytes, so the distribution is totally different. There is no NTPDase3 in the liver. So each of them are very specific and different localization. Another example is in the pancreas. So we have on the tops, on the panels here, we show the hydrolysis of ATP, ADP, UTP, and UDP, and the activity can be seen as a Ross color. And what we found is that the combined activities of these enzymes can be explained by the combined immunolocalization with NTPDase. Here are the antibodies. So NTPDase1 antibodies, 2 and 3. Again, as you can see, the immunolocalization is very different, but the combined hydrolysis of these can be explained by the NTPDase1, 2, and 3 in addition altogether. So this enzyme would be responsible for all the nucleotidase activity that we can see in the top panels. Just that you can locate that there is a endocrine system here, the Langerhans islet. And there is a Langerhans islet in each of these panels in the middle, as you can see here, 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 and here. And again, just to make a short explanation, is that the NTPDase, as you can see, are expressed in totally different type of cells in this tissue. And there is no NTPDase8 in the pancreas. So again, all the 4 NTPDase are expressed in different cell types. So now I'd like to come to the core of the presentation is to show biological function of these enzymes, to show biological real data. So on how NTPDase can regulate P2 receptor activation. So obviously NTPDase can prevent P2 receptor activation, but they can also facilitate P2 receptor as well. So the first example is on platelet activation. So when you put ADP to a platelet-rich plasma, this will aggregate platelets very well. Indeed, platelets express P2Y1 and P2Y12 that both respond to ADP, and both of these receptors are necessary to get an aggregation. If you block P2Y1 or P2Y12, there will be no aggregation. So you need both receptors to have a nice aggregation to form the clot. And in presence of NTPDase1 hydrolyze ADP, and there is no aggregation. In presence of NTPDase2 is not so good to hydrolyze ADP, and there is a nice aggregation, and the aggregation is even stronger than in the control. This is not surprising because after some time, the platelet release are activated. They release the content of their granules, which contain molar concentration of ATP and ADP. And then you convert the ATP from the granules from the platelets, and you can have even more ADP produced because of NTPDase2. And that would explain why we have a stronger aggregation here, because we use only 5 micromolar ADP for the stimulation. Then we tried ATP. ATP to platelet does nothing. In presence of NTPDase1, ATP is hydrolyzed to ADP, but ADP is also rapidly hydrolyzed. So the final output is there is no aggregation in presence of NTPDase1. But in presence of NTPDase2, we have a very potent aggregation due to the conversion of ATP to ADP. So that means that here, NTPDase1 would protect, would not allow platelet aggregation by blocking P2Y1 and P2Y12, while NTPDase2 would do the opposite. It would facilitate the activation of P2Y1 and P2Y12. And this makes sense because, as I have shown in another tissue previously, NTPDase1 is highly expressed in the endothelium. This is coronary arteries from the heart. And NTPDase1 is also expressed in the smooth muscle cells, as well as in the endothelial cells of the capillaries. NTPDase2, in contrast, is expressed by adventitial cells surrounding the coronary vessel. And in the heart, it is also expressed in capillaries, but not on the endothelial cells, on pericyte, on the pericyte that surrounds endothelial cells. So that makes sense. So in normal condition, and platelet sees NTPDase1, NTPDase1 removes ADP that is generated just endogenous ADP. So you protect the platelets. But when you disrupt the tissue, then you expose the platelet to collagen, von Willebrand factor. You release nucleotides because of cell damage. Then you expose NTPDase2 in capillaries and big arteries as well. And then you expose NTPDase2, and this makes sense. This is when you want to have platelet aggregation. So that makes sense for this. And this is just a confirmation by confocal. Now it's the time for commercial break. So I'd just like to tell you that all the antibodies that I will present in this talk, we generated all of them. And this is just to show that they are very specific, these antibodies. We have antibodies to NTPDase1, and in the control cell extract, they don't detect anything. So they are very specific. Same thing for NTPDase2 and 8. All of these enzymes are all of these antibodies don't detect only the one NTPDase. For example, they don't cross-react with NTPDase8 antibodies, don't cross-react with NTPDase3 that is expressed here, NTPDase2 or 1. So all these, and most of these antibodies are also species-specific. Some detect mouse and rat forms, but most of them are species-specific. So just to let you know that we have antibodies, and you can just ask them if you need. Antibodies to NTPDase, to NPPs, to 5' nucleotidase. So we have excellent antibodies. And just for you to know that often antibodies sold by companies, except for BU61, that is very good. Several of the antibodies sold by company don't even detect the right protein. So it's a concern when you want to make sure you have good data. So now I'd like to go to the second example. So this is, again, to show that NTPDase can control P2 receptor activation. NTPDase can also prevent P2 receptor desensitization. And again, I will show the example on platelet activation. I will show the effect of the absence of endothelial NTPDase1 on platelet activation. So what we did is just to look for bleeding time. And we just cut the tip of the tail, and this is exactly what we see when we do the experiment. It's beautiful. We really see that's what we see when we do the experiment. And what we were expecting at first, if we don't have NTPDase1, we should have more ADP surrounding. We should have more aggregation. We should have a mice that clot more rapidly. But we got exactly the opposite. So when we tested how much time it took for the bleeding to stop. For wild-type mice, it stopped in about 1 minute. But for the knockout mice deficient for NTPDase1, they did not stop to bleed. We had to stop them at 20 minutes. At 20 minutes, they might have died from bleeding. So the platelets don't work in the NTPDase1 knockout mice. To make a long story short, we observed that it was due to P2Y1 desensitization. So that means that if you don't have P2Y1, the endogenous level of nucleotides desensitize the receptor. That makes this not make the platelet not working. As I said before, you need activation of P2Y1 and P2Y12 to have a good aggregation. So having a P2Y1 desensitized make the platelet non-functional. So just a summary of this part on platelets and thrombosis. NTPDase1 is expressed on endothelial cells, and it prevents activation of P2Y1 and P2Y12 activation. In the same time, P2Y1 is also important to keep the platelet functioning well by preventing P2Y1 receptor desensitization. And then you have a third way to regulate P2 receptor activation, and NTPDase2 will, in contrast, will facilitate platelet aggregation by facilitating the presence of ADP that will activate P2Y1 and P2Y12. Now I'd like to show a second example on platelet, on the effect of NTPDase on platelet on P2 receptor desensitization. The model will be the effect on fertility here. So we observe after more than 800 liters that the mice that were deficient for NTPDase1 did not produce as many pup as a wild-type mice does. And the real effect is much lower than this because this doesn't account for this doesn't account for all the litters where it didn't work at all. So I will show just a couple of data to make a very long story short. But we found that the problem was due to male by in vivo ovocyte. I can't see my slide. By in vivo ovocyte fertilization, we found that the male were a problem due to the presence of the absence of NTPDase1 in the vas deferens. As you can see here, the male, when there is no NTPDase1, sometimes they can ejaculate pair spermatozoid correctly and have a nice ovocyte fertilization. Sometimes it works partially, sometimes it doesn't work at all. This all depends on the state of desensitization of the receptor. It's jam. Okay. So as I said, NTPDase1 is expressed on the vas in the smooth muscle cell vas deferens of the vas deferens. And you can see here there is high ATPase activity in wild-type mice, the presence of NTPDase1 in smooth muscle cells. In the knockout cells, in the vas deferens, again, there is no activity on smooth muscle cells, and obviously there is no immunolocalization of NTPDase1. So just to summarize all these some of these data, but of course we did several more experiments, but just enough to conclude that NTPDase1 is the dominant ectonucleotidase in vas deferens smooth muscle cells. NTPDase1 affects sperm emission via the regulation of P2X1 receptor activity. P2X1 receptor is absolutely very important for dysfunction. If you don't have P2X1, dysfunction just doesn't work. And in other words, NTPDase1 favors male fertility by preventing P2X1 receptor desensitization. So I had a fourth example for that shows that an NTPDase can prevent P2 receptor activation, but I think I'm over the time. So do I have 2 more minutes to explain this, or should I go directly to the final concluding slide?
Oh no, you have 2 more minutes. Yeah.
Okay, thanks. A nice guy. So the fourth example that I wanted to show is to show that NTPDase can prevent P2 receptor activation. So here it will be NTPDase8 that prevent P2Y6 activation. So just to show that NTPDase8 in the intestine, as you can see with this rust color again, NTPDase8 is expressed by the epithelial cells. So at the luminal surface of the intestine, and it hydrolyzed nucleotides in the luminal space of the intestine. And what we did, we generated knockout mice, and we found in the model of IBD, in the DSS model of IBD, that the mice deficient for NTPDase8 are very sick. They have colitis and lots of inflammation, much more than a normal mice, a wild-type mice, compared to the control. Of course, when there is no DSS, there is no colitis. So these mice are very sick in the absence of NTPDase8. And again, to make a very long story short, these mice, the NTPDase8 knockout mice, which are very sensitive to colitis, with a very high grade of colitis, this inflammation can be reversed by a dose-dependent concentration of P2Y6 antagonists. As you can see here, you increase the dose of the P2Y6 antagonist given intrarectally, and you can reverse the inflammation in these mice. Of course, we have much more data to support the implication of P2Y6 in this system. So just to make a quick summary of this fourth example, NTPDase8 is expressed at the luminal surface of the intestine. It hydrolyzes nucleotides in the lumen of the intestine. And by doing so, it reduces inflammation by preventing P2Y6 activation. P2Y6 is not known as a receptor that can desensitize, So obviously, an NTPDase can block the activation of this receptor. So that's a second example where an NTPDase can block P2Y activation. So the take-home message that I would like you to remember, if you understand this slide, you can forget everything I said today. So the first point is that NTPDase 1, 2, 3, and 8 are the most important ectonucleotidase and NTPDase to hydrolyze nucleotide at the cell surface. These NTPDase are expressed on different cell types, so you can allow different functions depending on what is cell surrounding, P2 receptors, and function of these cells. And these NTPDase hydrolyze nucleotides distinctly. Each of these enzymes will hydrolyze nucleotide distinctly. NTPDase can affect P2 receptor activation in 3 different ways that are presented in the next three points. NTPDases can prevent P2 receptor activation, especially those, of course, that have that can be activated by micromolar concentration, because the Km of NTPDase is in the low micromolar range. Of course, P2X1 that is activated by nanomolar concentration of ATP. I don't think you can block P2X1 with any amount of NTPDase, but you can easily block P2Y receptors, and you can very easily block P2X7 activation. NTPDase, the second way that NTPDase can affect P2 receptor activation is NTPDase can facilitate P2Y receptor activation, especially those responding to a DP and UTP can be facilitated by the presence of NTPDase2, for example. A third way that NTPDase can affect P2 receptor activation is that NTPDase can prevent P2, not only can, but sometimes are really absolutely necessary to preserve functions, as you could see with platelets and the vas deferens. You need NTPDase to prevent desensitization, and we showed here in this talk the desensitization of P2Y1 and P2X1. And of course, in addition to all of this, as this is normally ATP that is released from cells, and NTPDase together with 5' nucleotidase or alkaline phosphatase will produce adenosine for the activation of P1 receptors, which is also obviously very important for biological functions. So this is just to acknowledge the support and a few active collaborations. So a few collaborators here are presented here. And thank you.
Thank you, Jean.
Thank you very much, Jean, for the nice talk. And so don't feel free to ask questions on the chat. We will ask to the speakers at the end of the session. And the next speaker is Elena Adinolfi. And so please, Elena, very pleased to be with you today.
So I hope you can hear me.
Very good.
And see my slides. So I would like to thank the French Purine Club and the organizers for giving me this nice opportunity to present some of our recent work, which will be focusing on new advances on the role of P2X7 receptor in oncogenesis. As most of you know, the P2X7 receptor is an ATP-gated ion channel, which can mediate the opening of a small cation channel for sodium and calcium to enter the cells and potassium to efflux from the cell. And in this form, as channel, especially as calcium channel, it has been demonstrated in oncogenesis to promote tumor cell proliferation. On the other hand, the P2X7 receptor.
Can you enlarge your presentation because you are in the presentator mode? Thanks.
Oh,
you need to swap display again, Elena, like we did in the beginning.
Okay, let me see if I can
On
do
the
it
top,
somewhere.
on the top, yeah,
Yes.
yeah.
Yep.
Okay. Can you see the old slide?
Much better.
Yes.
Thank you.
Okay. So the P2X7 receptor, when exposed to higher concentration of ATP for a prolonged time, can also mediate the opening of a big unselected pore, also known as the macropore, and this in oncogenesis has been associated to cell cytotoxicity. In general, our and other groups' work has been proving in the past that P2X7 in oncogenesis is related to uncontrolled cell death, activation of angiogenesis, local and distant site invasion, metastasis, and in general, poor prognosis. The first subset of data I'm going to show is related to melanoma, where we analyze the expression of P2X7 receptor in a cohort of around 200, 300 melanoma patients, and we could associate overexpression of the P2X7 receptor with metastatic forms, which are interesting the distant organs distant from the primary tumor, such as the lungs, and other distal organs, such as the brain or liver. Therefore, we wanted to test if also in the animal models we could prevent dissemination of tumoral cells by administering a P2X7 antagonist. This is A74003. And as you can see, here is a dissemination model obtained by injection of SKML melanoma cells in the tail vein of nude mice. And we see the cells by intracellular luciferase emission. This is the placebo, and this is treated with the P2X7 antagonist. And as you can see, there is a significant reduction of the spreading of these cells at day 33 from injection. And this could be also reproduced in a fully immunocompetent model obtained by injecting, again, in the tail vein, P60 cells in the black 6 mouse. And you can see here there is a significant reduction in the metastasis. The metastases are clearly visible here in the placebo because are these black spots that are almost completely absent in the treated samples. We then asked which could be the mechanism underlying P2X7 mediated facilitation of metastasis. And we then think about microvesicle release. This is because it is really well known from cancer students that microvesicles and exosome can be favorable for cancer diffusion and metastasis. And we do also know that P2X7 activation can cause the release of microvesicles from immune and neuronal cells. And here is an example of macrophages stimulated with the P2X7 agonist BZ-ATP, releasing these massive amounts of microvesicles that we do know also contain, for example, cytokines such as interleukin-1 beta. However, nobody had ever demonstrated that also cancer cells, such as melanoma cells, could release vesicles upon P2X7 stimulation. Therefore, we tested whether this could be the case also for cancer cells. And these are, again, SKML human melanoma cells, stained in red with a stain for the cell membrane, and in green we have a quinacrine staining, which should stain both ATP and nucleic acids. So if we stimulate with BZ-ATP, also in this cell we get a massive release of vesicles, and as you can see, some of them do contain quinacrine, thus suggesting that they may contain ATP and also nucleic acids. So the following characterization we performed was to demonstrate that some of these nucleic acids are microRNAs, which are important in cancer, and are also metastasis-promoting microRNAs. So to analyze the content of microRNAs of our vesicles, we perform an experiment, a next-generation sequencing experiment, starting from 2 different human melanoma cell lines. These are metastatic melanoma cell lines, which do express P2X7 receptor. We treated them with 3 millimolar ATP, and then we collected microvesicles and exosomes by differential ultracentrifugation. Then we extracted the shorter mRNAs from these vesicles and analyzed them with the next generation sequencing, analyzing more than 2,000 different mRNA. And then we compared the levels of these mRNAs in the vesicles obtained without stimulation and with stimulation with ATP, therefore measuring the difference in the content of miRNAs. And its Vulcano plot shows the analysis for all the vesicular fractions analyzed and all the cell types. As you can see, there were many microRNAs which were upregulated here in red, or also in this scheme, following P2X7 stimulation, and some which were also down-modulated. And we get a massive difference in the microRNA content in each of these vesicular fractions. Therefore, we decided to take 3 different microRNAs, which we found upregulated in the NGS assay in all cell types and in all vesicular fractions, so both exosome and microvesicles, and in the mammal and SK-male cells. And these microRNAs are the following. Here is an experiment showing that we can confirm upregulation of MIR495, MIR376, and MIR6730 in all this vesicular fraction upon ATP application. And most interestingly, if we co-apply ATP together with the P2X7 antagonist A74003, almost in all conditions, we get a reduction of the secretion of these miRNAs. And this is of particular importance because we do know that vesicles are important in cancer, and we do know that miRNAs contained in vesicles are important to cause the spreading, select the metastatic niche. But there are no pharmacological option, good pharmacological option to block release of these miRNAs and to block release of microvesicles. Thus, P2X7 receptor could be a good target for this kind of therapy to prevent microvesicular release in cancer. We then prospected our 3 miRNAs into our cell lines, and we were able, with the scratch closure test assay, also to demonstrate that MIR376C is also able to reduce significantly the movement of the cells in the scratch test, therefore possibly favoring migratory activities. And as I showed you before, these vesicles also contain hinacrine. So we know they contain microRNAs now, but we wanted to test also if they contained ATP. And indeed, this proved to be the case, not only from cancer cells, but also from other kinds of cells. Here are vesicles derived from astrocytes, which do contain extracellular ATP. And in cancer cell, we even showed that not only this vesicle contain ATP, but they also bear mitochondria with them, so they can make their own ATP inside the vesicle. So the vesicles are for sure a way in which cancer cell or other cells interacting in the tumor microenvironment can release and cause an increase in the extracellular ATP. So it's not just necrosis or cell damage which causes release of ATP, but there is also an active release of extracellular ATP in the tumor microenvironment. So to study whether P2X7 receptor could also affect the levels of extracellular ATP in the tumor microenvironment, we took advantage of the PMLUC probe. This is a luciferase which is engineered to be expressed at the outer facet of the plasma membrane. So it allows to measure only extracellular ATP and not intracellular ATP. And because it's a luciferase, it can be used in a luminometer for small animals, and therefore allows for imaging of extracellular ATP in live tumor-bearing mice. So when we prospected our melanoma cells with the PMLUC and caused by injection cancer formation in the black 6 mouse, we can see that there was a significant release reduction of extracellular ATP levels in the P2X7 null mice as compared to the wild type mice. So we see less extracellular ATP in the knockout mice than in the wild type. Interestingly, this decrease in extracellular ATP correlated with an increase of the dimension of the tumor mass. That's meaning that possibly this decrease in extracellular ATP could favor tumor growth. Therefore, we talked about adenosine, and we talked that this could be due, this increase in extracellular ATP could be due by a reduction, an increase in immunosuppression. So a reduction in the inflammatory activity due to the degradation of extracellular ATP by ectonucleotidases. And I don't need to go in details about ectonucleotidases because we just heard about it. But I can tell you that in the tumor microenvironment, both CD39 and CD73 are actually studied as a main immunosuppressive factors, thus favoring the escape of the tumor cell from the immune system. Therefore, we analyzed whether in our model there was any change in the expression of CD39 and CD73 on immune cells present inside our tumors. And indeed, this turned to be the case, as especially CD73 was upregulated in most of the cells which were infiltrating our tumors in the P2X7 null mice. It was upregulated on Tregs, where also acted as a fitness marker, but also on effector T CD4 T cells and on the monocyte macrophage lineage. Interestingly, also CD39 was upregulated on effector T cells. So this data suggests that the decrease in extracellular ATP could be due not only to reduce secretion of ATP via P2X7 receptor, but also to increase activation of ectonucleotidases. This leading to a general immunosuppression in our tumor microenvironment. And indeed, this turned to be the case, as when we measure the serum levels of most of the pro-inflammatory and tumor-eradicating cytokines in our null mice, we could find a reduction of these tumor-eradicating cytokines. Of course, as I expected, we can see a reduction of interleukin-1 beta, but we also see a significant reduction in TNF-alpha, interleukin-6, interleukin-12, interleukin-17, and interferon-gamma in our mice, while on the contrary, TGF-beta, which is an immunosuppressive in the tumor microenvironment, was upregulated in the P2X7 null mice. So all this situation, the upregulation of CD39 and CD73 on the immune cells, and all this reduction of pro-inflammatory cytokines made us think about immunosuppression in the P2X7 null mice, which could be mediated by the adenosine receptor, as adenosine is the end product of degradation of CD39 and in particular of CD73. Therefore, we decided to check whether there was any alteration in the main receptor known to bind to adenosine and to be involved in immunosuppression in cancer, which is the A2A receptor, which you have been hearing also in the previous presentation. I don't go into detail. And indeed, this turned to be the case. This is an immunohistochemistry of our tumors growing either in the null mice or in the wild-type mice, and this is a staining for the A2A receptor. As you can see, there is an upregulation of the A2A receptor, especially on cancer cells. And we can see that this upregulation of the A2A receptor was mainly localized close to necrotic areas in our tumors. This was also shown in different specimens, and we could confirm it also by Western blot of the tumor mass growing in the P2X7 null mice versus the wild type. Interestingly, we can also measure, again by Western blot, an upregulation of the A2A receptor also in the spleen of these tumor-bearing mice, thus suggesting that the A2A receptor might be upregulated not only in the cancer cells, but also in the immune compartment in these mice, while in the liver, there was no significant upregulation of the receptor. So because, as I told you, we generally saw an upregulation of the A2A receptor in the close vicinity of necrotic areas on our tumors, And because the A2A receptor in cancer has been associated to hypoxia and vascular formation, neovascularization, we decided to stain for CD31, that is a marker for neovascularization in cancer, which was upregulated in the null mice as compared to the wild type. And this was also accompanied by an increase in the levels of vascular endothelial growth factors inside our tumor masses. So we then decided to see what happens if we block the A2A receptor, what happens to tumor growth. And so we administered this A2A antagonist SGH something, both in the P2X7 null and the wild type mice. And as you can see here, the antagonist was able to reduce, the A2A antagonist was able to reduce tumor growth both in the knockout mice and in the wild type mice. And interestingly, this was seemed able to normalize TGF-beta or reduce significantly TGF-beta levels in both of our models, but VEGF was reduced only in the P2X7 knockout mice. So to summarize, the P2X7 receptor is upregulated in metastatic melanoma, and its antagonist can reduce melanoma spreading in vivo. P2X7 activation causes the release of vesicles containing miRNAs, ATP, and mitochondria, and these vesicles are both microvesicles and exosomes. P2X7 blockade can alter the miRNA content of tumor-released vesicles, and this makes P2X7 a potential pharmacological target to reduce pro-tumoral roles of vesicles. The P2X7 receptor can also affect other players of the purinergic adenosinergic axis, as it can decrease the levels of extracellular ATP in the tumor microenvironment, increase the expression of ectonucleotidases CD73 and CD39, and can increase the levels, its loss can increase the levels of CD73 and CD39, and also the expression of the A2A receptor. So these two mechanisms seem to be related. This also, the blockade of A2A receptor also can partially revert the protumoral activity of the loss of P2X7 receptor, suggesting that the therapeutic strategy combined against both P2X7 and the A2A receptor could be beneficial in cancer. And at the end, let me thank all the people who did the work, in particular Anna Pegoraro for the work on vesicles, and Elena De Marchi for the work on the A2A receptor and P2X7 knockout mice, the funding agencies, Italian Association for Cancer Research, the TransCan program of the European Neuron, and Cost Action. And I will take my last minute to remind you about PRESTO Cost Action. PRESTO stands for P2X receptor as therapeutic opportunity. And if you are interested about our activities, please go to the PRESTO website or contact me to join the action. I know that many of you already are part of this action, and I thank you for actively participating. It is centered on P2X receptor, but we also accept in our conferences talk about the A2 and ectonucleotidases, so you are all welcome. And we have some slots still open for our upcoming activities. The next one will be a training school that will be held in Pisa from the 4th to the 5th of September. And following immediately after, we have a conference from the 5th to the 7th of September. So if any of you is interested in giving a talk and participating, please contact me. And then we will be organizing, as David said before, probably on next year, on September next year, the second European Purine Club meeting, which will be paralleled by a conference by the Cost Action. The interesting thing is that we get funds to reimburse your travel and your expenses, so the participation to the conferences is free, and also we have some funds to for students moving from one laboratory to another. So please, let's get in contact if you are interested in any of these activities. And thank you again for your attention.
Thank you so much, Elena, for this very nice talk. And now the session is open to questions for the different speakers.